This is Evo Devo. Oh, one thing I wanted to say, I have a correction to make about last time I did a video review, and since I'm taking these and posting them, I thought I'd say it while I'm about to start this, so I can just add it to the posted version, so if people are watching them in sequence, they'll get the correction. On molecular shape of view, I made the mistake of stating that an excited atom alone in the universe would not decay. That's not true. There is in fact spontaneous decay of excited states of atoms. I was thinking about the quantum mechanical, like the quantum mechanics picture where the electromagnetic field is still classical, and in that regime, that regime doesn't really work well with photon emission. But if you do it in quantum electrodynamics, it turns out that your atom, like the only stable state of a universe with one excited atom in it, is one where almost all the probability is that you have a de-excited atom and a photon, and the excited atom makes up a very low amount of the probability in the stable state in that universe. So I just wanted to clarify that. Thanks to a discussion I had with some folks on the Facebook page, implying that we can discuss physics, which is a nice Facebook page. So that erratum out of the way. Let's get into this one. You'll notice that this one starts where the last one left off. They're kind of a matched set, aren't they? In the last video, we zoomed all the way out to the human being. And now we're going to take a bit of a zoom back in and see some, just like stop at kind of a mid-range and see what's going on kind of in the informational point. Because once you've watched the molecular shape of you, you know that you're made of quantum fields and atoms and molecules and whatever. But what you don't know is how those atoms and molecules know how to do what they're doing. And you kind of get like a basic idea of this in your first biology courses. Like, hey, there's DNA, and DNA is like it's a code of some kind. It encodes for proteins, and the proteins make your body do things. And it's like, okay, cool. But then if you actually stop and think about like the immense complexity and fractal structure of the body and how meticulously it's planned out, and also the fact that every one of your cells has the same DNA, and yet all of them are doing something different, you realize that is not the whole story. It's not just, you take a library of DNA and you render all the proteins and then the proteins make you. There's computation going on in your biology to decide where everything goes and which cells should become what and all that. And it's partially epigenetics, as Melissa Gevin is saying. Epigenetics is kind of like, I feel like the way that it's structured is a little weird. Like epigenetics generally refers to like things like proteins attached to the chromosomes in specific ways that do specific things. So like certain parts of the chromosome will be packed up really tight, and that'll make it less likely to be replicated. Epigenetics also refers to methylation, so like little methyl groups are put on the DNA. And I think is it right that the methyl groups, do they activate them or inactivate them? I think they inactivate them. But Evo Devo is sort of another thrust of this, how your body controls. Histones are the proteins that wrap up your chromosomes. It's another thrust of this, and I learned about this from Richard Prum first, who was the guy I interviewed after I did the More Than Birds video. And he just kind of threw it up as like, he was talking about sort of his view of the origins of beauty and how he thought that sort of a new revolution in evolutionary theory was going to be the study of how minds and choices affect evolution once you get to beings that have minds and choices. And he sort of threw out, it's like the last revolution. I feel like it's going to be a revolution like on the scale of what we had with Evo Devo. And I was like, hold on, what's Evo Devo? I have to look that up because I was unaware of this whole thing. Let's see, methyl groups inactivate. They downregulate transcription if it's on a promoter. Okay. We'll try to explain promoters. I'm so bad at remembering because I didn't officially put the names of things in here. I'd be really bad at remembering what the names of different sort of like control regions on the chromosome are. Like what's the difference between a promoter and an inhibitor, and there's another one that isn't promoter or inhibitor. in any case, so I got into, I watched like a couple of crash course videos on the subject, and then I dove into this book by Sean B. Carroll called the Endless Forms Most Beautiful, the new science of Evo Devo. And when that book came out, I think it was fairly controversial, like how strongly it was stating the case. But in the meantime, it was 2006 that it came out. By the time I was releasing this, it was much more established. And then like after I read that book, so this video is essentially like a distillation of that book. It's really a book adaptation set to Despacito. So if you want to know a lot more about Evo Devo, I suggest you go get that that book by Sean B. Carroll, because it goes through everything that I'm saying here, but in much more detail. And then after that, I got after reading that book, I got a little more into the weeds to actually figure out things like I got really into like proving to myself that that transcription factors were actually like a I don't know about Turing complete, but like calculate, like what's that word for if you have a set of logic gates that can perform any calculation? Is it? It's not Turing complete, because Turing complete requires a tape, and like a little Turing machine. But anyways, you can do that if you have XOR gates. So I got really into figuring out if you can take transcription factors, complete Boolean base, yeah, that's it, and form a complete Boolean base with them. Which basically just means, can you make an XOR gate out of it? And it turned out, please don't reduce the whole Evo Devo to genetics and transcription factors. Did I do that, Ginger Rogers? Tell me what I missed, because I feel like that's pretty much what I took here. But it's like that's sort of the bottom level, but there's like the way that plays out is a lot of different things. But I don't know, Ginger, if you want to give me more context, tell me what I missed, because I don't know if I have a full view of this field or not. But even like what I got obsessed with as like, whoa, that's so cool, is the idea that transcription factors form a complete is it NAND forms a complete Boolean base, but XOR doesn't. Which one was it then? What was I doing? I don't know. I looked up like the simplest one. Maybe it was NAND, and tried to figure out how to make it with transcription factors. And I spent like a bunch of time on that. I got really crazy with it, and I posted a lot of Instagram screenshots of me doodling with like these like genetic circuits. And that led to a lot of these like, I don't know. I think that like there is actually this circuit, for example, actually does make sense. It would do what it shows here, at least in terms of like making this gene actually read out. So yeah, basically the biology has figured out a way to implement logic gates in this with these transcription factors, which I think is pretty cool. And we're gonna watch this video, and we'll go to more of it as we get going. Oh, so sad.
Hi.
I don't know why I'm always like, I have this need to lampshade the fact that I'm awkward. And you'll notice that I still do it when I'm reviewing these videos, but it was just like they do this like I thing at the beginning, and I was like, I can't pull that off, but I can be awkward. You'll notice that this was shot after everything else. I've got quite a bit more of a beard. So these are So let's see. There's what are these books? We've got Thomas Henry Huxley was like he was called Darwin's book bulldog. So he was kind of the guy who originally championed Charles Darwin. Ginger Rogers, give me some good sources, because I would love to like if I've missed a whole bunch, I would love to make another video on this. And don't put it in the chat, because I'll lose it. But if you want to tweet at me or put it on Discord, that would be really cool. I'd love that. So this is Huxley was like the sort of Darwin's champion, like the guy who really went into the arenas with people who thought evolution was terrible or evil or whatever, and and fought for the acceptance of, or like the sensicalness of evolutionary theory. And then this is Barbara Mcclintock, who was like chromosomal constitution of the races of maize. I called her. I don't think anyone calls Barbara Mcclintock BMAC, but I need a song to put in that spot. And I was like, wouldn't it be cool if like this, this like scientist who figured out all these ways that, like, she really focused on corn, as you can tell from, well, I mean, she did a lot of things, but this book was focused on maize and like sort of sorting out how maize and corn develops and how different parts of it are knocked out if you knocked out specific things. I don't remember super well what she did, but I just thought it would be fun if she had a rap name. It's like, yeah, BMAC and the boys. So there's Sean Carroll. And Stephen Jay Gould, Ontogeny and Phylogeny is like this gigantic book. I haven't read it, but he was. I think Stephen Jay Gould. I don't know how much he uncovered the original Evo Devo stuff, but he was really pushed. He was one of these people that was never satisfied with the current state of things. Right, like one of his big ideas, which it's sort of, I don't know. I think people still argue about it, is like the punctuated equilibrium idea. But he for a very long time had this sense that like the understanding of evolution was incomplete. I guess that like even though we had kind of like the new, like I don't know, I guess like the standard model or whatever you want to call it, the modern synthesis, I guess how it's referred, like Darwin's evolutionary theory plus like DNA and the story of how DNA gets turned into proteins and stuff. The modern synthesis. He had he kind of didn't believe that was the whole story. Like, and I think he was trying a whole bunch of different things to sort that out. And Evo Devo ends up being one of the things that sort of solved that problem, just in terms of how do you actually, like, how do you generate enough information, enough complexity within the bounds of evolution? Like, you can't just have DNA that makes proteins that then go and specifically cause some sort of phenotypic change, because there's just there's too much complexity and repetition, and like you need an organizing system for it, and that's sort of what Evo Devo has turned out to be, is like uncovering this, like, these bits of DNA and proteins and whatever else that don't actually act as, they don't phenotypically act, they act on other genes, so they're feeding back on each other in complicated ways that are basically the equivalent of logic circuits. So Stephen Jay Gould, a real pusher in the field, I mean a giant in the field, but yeah, we got him, and then we've got Davidson. So Davidson and Peter. I believe Davidson is now deceased, unfortunately. Isabel Peter. I wanted to get her on my podcast, and she actually said yes, and then I dropped the ball on it. I still haven't talked to her. But this book, I managed to get my hands on a copy of this when I was preparing Evo Devo, because this really goes into like one of the main things they were trying to do was to work out like gene by gene all the way up from the first cell. I think they focused on a sea urchin, and they worked out like for the first, I don't think it was all the way up, but it was like it was like for the first 30 or 60 days, like exactly what's going on, like what is triggering what's feeding back on what, so let's see. Ginger Rogers. The general rule is that Evo Devo is how evolution influences development and how development itself evolves. Yeah, I think Ginger. I think we're talking about. Similar things. Because the way, like, it's not like your body is. Like once you have organs in place, they're sort of set. So it's not like a full on like Turing complete computer type thing where like you're always free to reprogram and change around. I mean, there are some layers of that. Like, for example, clownfish by their social setting will like their brain will completely feed back and change their sex, and it's pretty crazy. So there's a lot of reorganization that can be done, but like once you differentiate a cell, it's pretty hard to undifferentiate it. So that's why it's like it's more like a calculation from a calculator than like a Turing complete program. But what that is like, so development is this unfolding of kind of like this calculation of taking a little bit small amount of information and using like repetition and inhibition and promotion and stuff. Like if you've ever seen like a domino computer, like once the dominoes are all fallen down, it's done, and it's going to stay that way, but also it's done something really complex. And so that like the development is driven by this kind of calculational system, and that has really big impacts on how the system can evolve. Because, for example, really early on, you might have one transcription factor or like signaling protein that's doing like 10 different things. And if you change that, it's going to change 10 things in development. It's not going to change one. So there's all these things that are correlated, or that have multiple functions, right? Because we can have one process initiated by like 3 proteins and another process initiated by just 2 of those proteins, or those same proteins, but like down the line when there's another thing in place. And so these control sequence, like these control elements are really multifactorial, which makes it really hard to change them, which means that like early development is really locked in place. There's not a lot of ways that you can change the first 30 days of life without changing everything, right? And like breaking your thing and probably making it super hard for it to survive. So for example, the early development of all animals is like exactly the same. Well, not exactly, but like in larger and larger families as you get down to the like the earlier and earlier stages. We all start as a blastocyst that gastrulates, and then like if you look at the early embryos of like fish and humans, they pretty much look exactly the same, because all of those processes are like so fundamental that you can't particularly mess with them. So that's a real constraint on like what we as a whole clade of like chordates, like animals with a nervous system that goes down our spine, could ever become. There's kind of this valley that we've fallen into that we're fixed on. Yeah. So let's continue on. The other thing with that is that like if you can find a switch early on in that process that does work, you can have a rapid, sudden large scale change in the organism. So for example, this thing that probably happened with like centipedes. They didn't have to re-evolve every new segment. They just had a template for a segment, and then you flip an earlier switch that adds one more, and all of a sudden, instead of a normal insect or a normal little arthropod, you start stretching out and stretching out and stretching out. It could probably happen over evolutionary time very fast, because you're doing one mutation like really early on in the sequence that has these cascading effects. So it's like a constraint, but it can also be an amplifier. Anyways, so here's our cell. Our cells start off. This is the cell from Molecular Shape of You, right? I took the exact same the exact same one and just copied it and changed the color and stuff. But our early cells, the egg cell could become anything, but it doesn't become. Your early stem cells, they can become anything, and then they have particular programs initiated in each one of them that differentiate them. Decide on 1,000 fates. Let's see what we got here. Gamete, neuron. Your cells are the variation in cells is crazy. Like especially the cone cell. It's just freaky. Someone's asking if you can have a NOT gate in domino logic. Yeah, sure you can. You just have one path of dominoes knock over another path of dominoes. Like knock it sideways, knock a domino out of the way, and that stops the signal. Did you ever figure
figure how they know.
Black box. How do we do it? Whenever this is a real physicist term, because whenever physicists don't know, I mean, I don't know if it's just physicists, but whenever scientists don't know how something happens, but also it doesn't matter. They're just like, well, it's just we'll put it in a black box, and it's like someone handed you a black box and you have no idea what's going on inside it, but you can put an input in one side and it comes out the other side and you're happy. So it's like metaphorically me pointing out that for a long time this differentiation was a black box.
Figure how they know. Be math. We are built of modules combined in a planned out way.
This animation took so long to do. Like I usually, for most of these, I went and found like pictures of these creatures, so like a picture of a centipede or a picture of a human hand. But then I did all this, this tracing of the thing myself. I'm not that good of an illustrator. Like I wouldn't be able to draw this freehand. I don't know what a centipede looks like. I really had to go and get images and trace them over every time. And then all these little like bloop 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 bloop. Yeah.
Each new piece must be
told. This is like a mouse skeleton here.
Where to go? Oh.
And then you can imagine like placing each one of these things randomly around, and then going in and keyframing each one of them to, like, you start with the full mouse. You work backwards for this one. You start with, maybe it's a rat. You start with the full rat, and then you take it apart piece by piece and like mouse it, scatter it around everywhere, and then you have to keyframe it all to come back together when you want it to. It's a labor of love. Luckily, I was on a bit of a high from having molecular shape of view do so well, and I was like, I'm gonna make this even better. Yeah. Your skull looks ratty to me. Yeah, it's probably a rat. You're probably right, Jan.
These must be told where to go. Oh, now there's a science helping us.
Aha! Hox genes. Okay, so Hox genes are this. I mean, you'll see them more in a minute. So maybe I'll stop it when we show all of the different Hox genes with the different species, and then we can explain it better.
Understand how our cells encode this architectural plan, signaling each other with genetic tools. Oh yeah, wow.
I really like
Phenotype the interface for mouse and man. Genotype the files and the sub programs. What then are the switches, circuit boards, and boot code?
Does that metaphor make sense? Like that. So the idea is, like you've got, I don't know if this metaphor makes sense. Somebody tell me. My idea was, look, you've got like the visible features of an organism which interact with the world and that you can interact with, and that's your phenotype. And similarly, so you've got something like a user interface on a computer, and then so then you've got that. That's like layer one.
Genotype the files and the sub.
And then like the genes are like, they're the equivalent of here's like the raw files that go into the programs that generate the interface and whatever else. But what's the program itself? Like, that was what I never understood. Like we have the input files and we have the output nice thing, but what's the computation? How do you turn a bunch of DNA into a mouse? And the answer is all of this computation.
Programs.
I do like this. Like I've got a point mutation that says file corrupted. I think that's I don't know. I forgot about that. I color good. This is how I always name my files. I'll make a file that I'm like, oh, this is for sure the best one, and then this is done, and then I'll discover some mistake, and the next one has to be saved as something else. So it ends up being like good, and then like I'll do this in my videos, like Evo Devo good version, Evo Devo definitely good version for real. Yeah. Why is it an exe? Because it's a user interface. It's not. I color final 3. Yeah, that's how we do.
Programs. What then are the switches, circuit boards, and boot code? Evo Devo. Looking at the logic in the ways that we grow. Every gene.
Yeah, there's a good morph. So you can see there the early phase of. But this is an early human embryo. But it doesn't really look particularly human, does it? It's got a tail. Kind of looks like a beluga whale. as close as I could decide. But if you look at an early embryo of a mouse or a sheep or a fish or whatever, they're going to look pretty similar to this.
Logic in the ways that we grow. Every gene directed by a signal key code. Proteins that can activate, enhance, or veto.
Okay, so this is the real crux of it, right? So the one thing that I didn't really get into here, and to be honest, I'm still a little shaky on, is how, like, in right down at the level of physics, how mechanistically this works. So the idea is that you've been told in your biology class that you've got a gene. This is your gene, right? So what usually happens is your or RNA, or what is it called? RNA polymerase comes along and it attaches here, and there should be like a promoter region here that I didn't really put. It comes along here, it reads your gene, it transcribes it into RNA. The RNA gets pushed out of the nucleus, and then it gets translated by ribosomes into proteins, and the proteins are little machines that go off and do things. So that's what you've been told. But what's also happening is that before, like, upstream from the gene, it's usually upstream because it's close to this. There's a region here called the I think it's called the promoter, where the RNA polymerase needs to attach, and what can happen therefore is that, like, the folding of this nearby region of chromosome can determine how easy it is for RNA to attach to the promoter. And so this could be a region that normally, if there's no protein, if it's not, so these are transcription factors. These are supposed to represent these little proteins that are made that are good at finding specific regions of DNA that have specific codes on them. They'll find it and they'll attach to it. And so then those proteins can go and also attach over here to the promoter region and mess with it in particular ways. And like all the promoter regions are roughly, I think all the promoter regions are roughly the same, which means that this is modular, right? So any any one of these different proteins that can mess with how easy it is to transcribe this can be put onto any gene and it'll sort of work. And so maybe this one folds over and attaches on here and actually completely inhibits the promoter, inhibits the RNA. Let's see, Ginger Rogers is in the chat helping me out again, because my view on this is the promoter is usually the area where the TFs and the polymerase attach, transcription factors and the polymerase attach. Yeah, promoter by itself works at a base level. The enhancers are other regions that help the promoter to recruit more transcription factors. Okay, so yeah, these ones, because your DNA isn't actually nice and just like this. It's always like jostling around, because down at that scale, like I say in the nanobot video, like it's like a hurricane, like everything's bouncing in different ways. So these regions of nearby DNA are always like jostling in and out of contact with each other. So you might have this one attach here and inhibit all the RNA polymerase from coming in, and that'll turn off the gene. Or you might have this one that if this protein is attached to this particular area of genetic code, this protein will attach on, and it'll have something that attracts nearby RNA polymerases to preferentially take this one instead of others. So you by exposing this segment of DNA to specific proteins, you can choose how much of this protein gets made. And the fun thing about it is that this protein could be another transcription factor, right? Like it could be, like, the bottom level is that this will just be a protein that actually goes and does something, right? It'll be like melanin or whatever. So you have something that either turns it on or off, and that determines whether in that particular cell that protein is active. But it can also be a transcription factor, which then whose job is to go and bind to another DNA region like this and do something else. So this is where you start to get into like logic gates type things, where this one in particular is acting as a knot for this signal to propagate and do something else. So maybe this will affect three different things. Maybe there are three other genes that this transcription factor will attach to and make that happen. And so suddenly each one of these will determine like this one will inhibit all three of those downstream actions. This one will excite all those three upstream actions. If both of these are on, then it depends on the particular mechanics, but you can have things like this is it only works if both of these are, if no, that's not true, but usually the inhibitor takes over. If the inhibitor is there, then it just clamps down on it. So it's like this one would make this greater, except if the inhibitor is there, and that's how you get all these weird logic gates as combinations of these. So when we get into the next image, maybe I can point out some examples if I can even decode them still.
Evo Devo. The signals are controlled by other genes that
Yeah, so for example, here's kind of a diagram of what we were just seeing. So all those three transcription factors on the previous one are made by their own genes that have their own, promoter and inhibitor regions attached to them. So supposing that these genes are all turned on, they're all feeding into this one at once. And so that's what you're seeing here. They're, right now, all three of these are attached. The inhibitor is going to take over. So nothing's going to get output of this particular gene because that inhibitor is just going to block everything off, I think. Or at least like these things are rarely fully Boolean. Like there's usually some like thermodynamic thing where it's more or less instead of all or nothing. But veto, yeah, I had activate, enhancer, veto, right? This one's kind of like the veto one. So there you can see I would have had something come off here. If this were working, I would have had a little train of protein coming off here and doing something else, but it didn't. It's vetoed. But let's see what happens if we add some more. Okay, so let's see what's going on here. We've got these two inputs coming in, and so the way to decode these is these ones are essentially will activate, and these ones will inhibit. So right now in this circuit, we've got these two activating. So here's what this circuit looks like. Like your gene can only really code for an inhibitor or an activator. Like it's not fully like logic circuits where you can have the circuit go one way into a NOT gate and another way into into an off and another way into an on. It needs it needs to go through a second gene to make that transformation. So each one of these is either coding for like a promoter or an inhibitor. So this one is feeding into this one and turning it on, right? And then that means that even though, so right now these two signals are on, this one's coming in, but this one is, because it's being fed by this one, is also feeding into this one, which is shutting it off again. So there's nothing coming out of here. This gene is turned off because the net effect is the veto from this one. So then the only thing coming down from this layer is this black one, which is also connected to all these three. So what's this going to do? This will activate this inhibitor here. So we've still got all three going into the red because it's activating this one, and this one. And this gene has been turned off, so there's no suppression coming into these two. So all these three are running. And so from this layer, all three of these signals are coming through. So we've got the activator, the enhancer, and the veto coming into here and turning that off. So nothing's going down. And so it turns out now in this diagram, this red one that we have been seeing all along was an inhibitor. Its job was to prevent cascading on the downward level. So we've turned off the inhibitor, which means that we are in fact exciting these genes down here. And this one in particular is just feeding over into that one, which will excite it. And as a result, we get an activation from both blue and this sort of brown color down into here one. And the end result is that we have this little, this is going to spit out, this is like an end of the line protein that's actually going to go out and do something. But if we had changed any one of these inputs, like I haven't followed it through with the other ones, but suppose we had changed this input to be on. If we had all these three inputs on, then this one would inhibit this, which would fail to inhibit this, which would inhibit both of these two. and only activate this one. So this would go down here and it would activate that, but it would also activate this, which inhibits this, and nothing would come out the end, right? Why does this need such a long explanation? I don't know. I just want people to follow through. If Brendan, if it's not your thing, I don't know. You don't have to watch it. But I think it's cool. Okay, so that's the whole early Drosophila development chain, which maybe I can explain to you. So Drosophila is a Drosophila is the model that a lot of this stuff was discovered in. It's actually a bit of a weird model because it does this very strange thing at the beginning of its development where it has this large, I can't remember what it's called, but it's this large enclosure where all of these, like, this isn't individual cells. So like usually what your cells do, they'll start with a single cell and they'll divide and divide and divide and make like a blastocyte, which is like a hollow sphere of cells that then do things and differentiate. But they always, a syncytium, that's what it's called, or how do I pronounce that? Syncytium? I don't know. In any case, because but here it's just like there's one membrane surrounding all of it, and these are just naked nuclei floating around. Syncytium, sensation? I don't know. You can look it up. Syncytium. Okay. So it's got this one. I think this is bicoid. This is a single transcription factor that comes from the mother, like it's already there in the egg, and it's just diffusing through this region of the syncytium or whatever you call it, syncytium. And because these are bare nuclei, this transcription factor can just diffuse through it and enter all of them. So it forms this gradient. So that's the first layer, is that you've got a gradient of sort of like maternal signaling protein going through all of these nuclei. And then you get to this phase, which is that you have two different genes that are inhibited by, they're activated by a certain level of this maternal gene, but they're also inhibited by a certain level. So they'll only be activated at particular places. So this one that I've put in green will only be activated by sort of this front end where there's a lot of it, and then it's not sufficiently activated after that to turn on. So it's like it's changing it from a smooth gradient into kind of like a yes or no, where it's either activated here because there's a lot, not activated here because there's not too much. But this red one is actually disactivated by there being too much. It'll be inhibited by there being too much, and also won't be activated enough if there's too little. And so there's only this nice little strip that gets differentiated. So now all of a sudden, instead of the one region, we've transformed that into 3 separate regions that can be differentiated. And then from there we get this. I can't remember how it is that this goes into the pair rule. These might be going on at the same I think these are going on at the same time, where there's this dynamic process that it inhibits or excites these stripes in inhibits or excites these stripes sort of along the whole thing. But I can't remember whether that's, I think that's also from the maternal gradient. I could be wrong about that. But it's one of these two gradients that it's like you've got these very particular genes that only correspond to a very specific bands, in the same sort of way that the gap gene did it. And then at this point, this thing starts to do weird shapes. I think at this point, at some point, these nuclei get enclosed by cell membranes. It kind of the cell membrane like gastrulates from the outside and kind of like pulls them all into little cells. So like now these nuclei are not just nuclei floating around. They've all had like cell membrane envelop them. They've become real cells. So then after that, you can't use gradients anymore. You have to work with what you've already got. I'm not sure if that's happened yet, but it's harder to do gradients with. You can't activate a transcription factor. You can't activate a gene with a transcription factor that's just like floating through the whole thing once the nuclei are enclosed in a membrane, because the transcription factor can't get through. So then you need to do what human beings and most other things do, which is that we have a signal protein that attaches to the surface of the membrane, and then that signals something inside the cell that is a transcription factor to go into the nucleus and do something. It's a bit of an extra process. And Drosophila have like, the reason why Drosophila can multiply so fast, which is why we like them for research, is that they've skipped one step of that signaling process, which means the signaling can happen really quickly. And so you can get like from a baby Drosophila to an adult in like a week. Okay, let's see what's going on here. What did I miss? So we got, okay, maternal, and then polarity is where each of these, like based on these stripes, you can double them based on basically the polarity of these like pair rules, like whether you've got yellow pink or pink yellow. You can differentiate those stripes, so you can get this polarity thing that turns on in double the amount of regions that this one turned on in. And then I don't know how all of that goes. I don't remember how all that works together, but it's like a combination of all of these different signals comes together to turn on these Hox genes, which are like a striping that differentiate the broad different regions. Let's see. Ginger Rogers says, due to the position of the cellular receptors, some cells signal front segment, some cells signal back segment. Sort of a feedback that enforces the division. Not sure, Ginger, what that refers to, because I didn't see it in time. But yeah, so now we have these Hox genes that are like, it's not so clearly striped here. Like I think there's a few regions where there's like overlap where 2 or 3 of them are turned on. Like you saw that back in, because we have the exact same Hox genes, and you'll notice in this one, in the rat model, there are some regions that like in us in early development, the Hox genes are turned on in sort of like overlapping regions. Like here's 11, 10, 9, they're all kind of overlapping together. But yeah, so we got now it's striped, and that will determine the different, like how these different parts develop from there on. The Hox genes are really the masters of that. So this Hox, I'm not sure if it's Hox 1. I think it's Hox 1. Actually, Hox 1 might be, yeah, Hox 1 is the head, and then like the head, and then that purple region became just like the little proboscis thing. The yellow region becomes each one of these for the broad different segments. So pretty cool, right? And like each one of these is actually, like it's not like the process ends there, that you get the Hox gene and then you magically have a head. That gene is then triggering a whole cascade of complicated calculation down the layers, just the way that this first one did for the broad segmentation. That'll turn it'll turn each one of these into a bunch of different regions that can differentiate and such. Okay, so these are, I can't remember what all these are called, but these are like the scientific name abbreviations of the ones that are found in flies. And these same ones are found in human beings and in mice. But something interesting happened. At some point between the point where we split off from flies, our whole Hox gene segment got duplicated in our genome, and then it got duplicated again. So we have, whereas the flies and like early worms and stuff, like this would probably be like our common ancestors, like this worm thing, right? It's like 550 million years ago. The flies got it once. We got 2 duplications, which allows us to have some more it just allows us more possibilities, right? Because once you duplicate these Hox genes, then they're free to mutate separately. You can keep you can keep all of the functions of this gene because you've still got one complete copy that works, but then this one can go off and start changing subtly and performing different functions, and they can get so far that they're like doing completely different things, and all of a sudden you have this burst of complexity that's possible because of this one macro scale adaptation, or this one macro scale, accidental mutation. And then the other thing that seems to have happened is that our last Hox gene in a couple of these regions got duplicated a couple times down here too. So we've got like extra tail segments we can work with. That's generally how we get, like, if you hear creationists asking, how do you get new information into an evolving organism? That's basically how it's done. You take old information, you duplicate it, and then you start changing it, and all of a sudden you've got twice as much information. So that's what happened here.
The Paleozoic era, baby. In a crucial pathway, changes tend to get torpedoed. Where they go, calamity goes.
Yeah, that's what I was talking about before, right? If something's really important in early development, it's affecting lots of downstream processes, you change it and just like nothing works. But if you duplicate it, then you can change the other one, and this one will still run all its functions, right? I had fun using this. I don't think this is a real circuit here. I was just like sort of making a hypothetical model involving this little mouse. But I had fun using the warping function until I felt like the mouse was suitably decrepit looking.
Where they go, calamity goes. As the cyclonic shield.
Hey, how's that? How's that for a skill? This is the only video that makes my that freaks out my niece because because of this particular image. I love this thing. I think this particular cyclopic sheep, I think, had one of its it's where'd you get this picture? I don't know. They talked about it in the Evo Devo video, and then I just looked it up. But what happened was, if I can go over to here, we've got a thing called hedgehog, which actually like is expressed in the middle of our foreheads. It's one of the hedgehogs. It might be Sonic Hedgehog that differentiates our head into two halves. It splits our brain into two lobes, and it like forms this line around which we're symmetrical. And sheep, there was some particular, what's it called? It's some sort of it's some plant that actually acts as a transcription factor. Like there's a protein in it that can act as cyclopamine acts as a transcription factor that inhibits hedgehog. And so if you don't have it, you give birth to a lamb where the two halves of its head never split, and it just develops one gigantic eye in the middle of its forehead. And also, like, they usually have this like weird proboscis looking thing sticking out of the top of their head. It's really bizarre. And it all comes from a change in one inhibitor of Sonic Hedgehog protein is robotnikin. Goodness. We'll talk about naming conventions of these proteins when we get to them. But yeah, it's like one little change, and there's the result. It's crazy. Downstream of an important molecule.
See down they cascade like a domino, like you and I, Drosophila.
This is a really fun verse to write, just like the domino. It's weird to do parodies of a song in another language. This is the first one that I've tried. I've attempted some K-pop songs, but I've never like succeeded at finishing one. So maybe one day one of those will exist. But this one has such a good like, and I don't know what the actual original words are in Spanish, but like as soon as I realized that Domino and Drosophila work together, this is a fun verse to put together, is all I'm trying to say. So let's see.
The path that makes us optical was laid a long time ago. Back before we blew up the Cambrian like a bomb. Now my eye protein can make you see out of your bomb and hedgehog and it's relevant.
So that's true. I can't remember. Is it someone in the chat told me what the name of that protein is, but I don't remember it because I didn't write it down in here. But it's the protein that encodes for like these set of these set of cells is going to be an eye. PAX6, this set of cells is going to turn into an eye. That hasn't changed. It's so fundamental to the developmental process of all beings with eyes that if you take the one out of humans and you put it into Drosophila, it'll trick their cells into forming an eye, because it's so hard to change a protein that's that fundamental like that. If you change it, you don't form an eye, and then you do not survive. I mean, like the downstream processes are completely different. Like, because these, both our eye and the fly eye, they come from way back in the day. This little worm that probably had like a few light sensitive cells that were activated by this protein. And after that, it went in all sorts of different directions. It became these crazy Cambrian monsters, and it became the early chordates and whatever else. But the fact that signal is the first signal that leads to eye development, that's now fixed. You can change the downstream program all you want. You can have the whatever the transcription factors bind to, they can bind to completely different proteins that do different things, and they can be mutated beyond recognition. But that initial signal is so fixed to make eye that it can never change. And so if you put that into a fly, it's going to get you put it into the butt of a fly. I mean, you can't put it into an adult fly. You put it into like an early fly larva, like a fly embryo. That set of cells will say, hey, that's our eye protein, even though it's the one from humans, and they just grow an eye wherever you put it. Which is, I saved us from the actual pictures of those flies that have, because people have done this experiment, those flies that have eyes on weird parts of their bodies, because it's also fairly disturbing. But I just did a cartoon of it. So here's what happened. There was one protein called hedgehog, which is I think it's one. Is it? It was one that was first discovered in flies. It's not a Hox gene. It's a sort of later transcription factor, it's not a transcription factor itself, is it? It's a signaling protein that signals the cells to activate transcription factors to do various things. But the one they discovered in the fly, they called the hedgehog, because they would always name these genes after whatever happened to the fly if they knocked out that gene. And when they knocked out that gene in the fly, it developed all these like spiky, I don't know, like spikes and weirdnesses about it. So they called it hedgehog. And then when we looked at human beings, we discovered that hedgehog had been duplicated. So we have 3 different copies of hedgehog. Like this one, like, and so people needed ways to differentiate them. So we called them Indian hedgehog, desert hedgehog, and since we had run out of types of hedgehogs that actually exist, we called the third one Sonic Hedgehog, because why not? Yeah. Okay, so that's they must be activated. If they're set up in a gradient like this, then they're probably activated by those polarity genes on each of these regions, and then they spread out, and they allow each of those segments to be further differentiated based on the gradient, the same way that the original bicoid protein did it for the broadfly. Also, I apologize that this looks fairly phallic, but that's just how fly embryos look. What do you want from me? So yeah, so this is how we get differentiation, or like in the head, Sonic tells you, or Hedgehog tells you how to separate out into 2 lobes. In the hand, it diffuses out from cells that secrete it in this part of, so like the hand bud, and it based on those gradients, it tells you which of those cells are supposed to go and form fingers and which ones aren't. So it's a very multifactorial protein. Those are just 2 examples, but you can see that you can see how knocking it out with something like cyclopamine could cause a huge problem. Unfortunately, Brendan, I did not use zebrafish or sea urchins at all in this video, so I'm sorry to disappoint. Zebrafish are a riot. Okay, so here's how hedgehog works. So once you've got membranes around your cell, you can't just directly diffuse proteins through your whole body and have them directly impact cells the way you could with the Drosophila in the early days. You got to have a more complicated pathway. So Sonic Hedgehog will attach to this patched 1 protein, which will then inhibit this smoothened protein, which are both membrane proteins, because proteins can also, attach to other proteins to inhibit them or make them do things or whatever. Is it inhibit? Am I saying inhibit here? It inhibits something. I think this one causes this one to send. Does it go in itself? Does it come in through the membrane, or does it send something else through the membrane into the internals? I'm not sure. Anyways, some signal has to get transferred from the membrane down into the cell. So you've got this GLI protein, and this is actually the transcription factor that then goes into the cell and attaches to something and causes proteins to be expressed. So it's a weird set of interactions. Isn't that the G protein? Which one? Is this the G protein? I mean, it's GLI. I can't remember what GLI stands for. Yeah, it's been a while since I drew this and I made sure it was right, but then I don't remember really what it means now.
Logic from Devo to Evo, adult and embryo.
Yeah, so this is what what was your name? It's talking about with it's the ways that development affects evolution is the broader scope of Evo Devo, because you could I'm mostly talking about how development, that's the question I'm posing, is how does development happen? But you can imagine this has two huge implications on how things can evolve. Any system that it's very different changing a bit of a protein that's just going to do something versus changing a bit of a protein that's going to have 1,000 downstream effects on other proteins.
From Devo to Evo, adult and embryo, mostly don't evolve in the genes of the genome. Save for the mutation, aim their regulation, keep the building blocks and swap their activation. From Devo to Evo.
Yeah, so that's the other thing, is that this gives a way for the whole organism to evolve without changing any of the proteins at all. None of these gene regions need to change at all, but if you can take one of these little regions and stick it onto a different gene, so you can activate them in a different sequence, it's a way where all the proteins get to keep their own function, but all of a sudden you can have a totally different program being implemented with all of those functional proteins. And so it's this sort of quantum leap in how you can evolve. It's like, instead of having Lego where you have to redesign the bricks every time, you just have bricks that you can swap around and build cool things with. So we would probably not have our modern day species if we hadn't figured that out.
For the mutation, aim their regulation, keep the building blocks and swap their activation. From Devo to Evo, parts of alter egos, homologs evolve from repeats in the scheme.
Yeah, so we've got this. The other thing this allows you to do is that if you have some sort of base segment, and you duplicate it, same as when you were duplicating the genes, you're sort of free to evolve. You're free to evolve them differently. So back in the day, in this sort of thing, Darwinic or whatever, this is like an early Burgess shale type arthropod, where it basically has one type of appendage and just these simple repeated segments. So it evolved a segment, and then it evolved. It added on a few different regions, and Screaming Tim right there. Are we pointing out where the Screaming Tim emote came from? Yeah, it's somewhere in this video. I knew it was. Then you're free to possibly what happened was that all three of these Hox genes were the same one, and then they got duplicated down the line. But either way, you can if you've got multiple ones activated, you have the freedom to start turning them on and off as you evolve. And then once there's a different activation in place for each one, then you're free to mess with the program for one without affecting the other one. Does that make any sense? I'm not sure.
Evo, parts of alter egos.
But yeah, so like in a lobster, the antennae and these like limbs here and the claw and these little claws and these things at the end, they all probably came out of the same template. It's like some sort of basal appendage, which is cool because what it means is that the program for, like, if you have one mutation that changes the early signaling in here, then your lobster, instead of developing a claw here, will just go along that different path, and it might spit out one of these little things or one of these little things. Or in the case of insects, if you add a single signal to the part of its body where it usually grows antennae, it'll just go along that signal chain and grow legs out of its face, which is what we're going to see.
Homologs evolve from repeats in the schema. Switch a couple bases in the proper places. You'll be watching flies grow legs out of their faces. Oh yeah.
Yeah, that's the other reason why this freaks out my niece. I really went with the shock here. It's just, look at that. It's like legs going right out of its face. I don't know. It's just cool.
Evo Devo. Stick around for modern synthesis.
That's the first. The modern synthesis was like Mendelian genetics plus Darwinian evolution.
Devo. Stick around for modern.
Mendel. You guys know about Gregor Mendel? For those of you who don't know, Gregor Mendel was like a monk in the 1800s. He was just messing with pea plants and figured out the thing, this stuff that we know now about dominant and recessive genes. And then Darwin came along and didn't know about Mendelian genetics, and did his whole theory of evolution kind of from first principles about things that replicate with differential rates of replication. And then the modern synthesis was realizing, hey, there was this monk called Gregor Mendel that figured all this stuff about genetics that we totally forgot about, and combining these into like the actual mechanism by which beings change and evolve over time.
Synthesis, the sequel. Only by combining can a new
And then this is the like developmental biology for a while. For it was like a whole other thing that was developing. This is what Barbara Mcclintock was working on, like understanding how it is that how it is that you go from single cells to organisms and that kind of stuff. But Evo Devo is putting those together. It's how does that then feed back on what evolution can do and what sorts of things it'll get. Am I saying this? Song has a ton of depth, averaging 30 minutes explanation for each minute of video. Yeah, we're gonna run out of time, aren't we?
regrowth, evolution and development, amigos. Evo Devo. Signals trigger patterns of complexity.
Oh man, these eye spots. So this is another thing that's caused by gradients. It's very similar to the way that the gradients in the early Drosophila formed, like we're able to signal off and on and form these like striped regions. This is how you see it actually play out visibly, is that this is exactly how these butterflies' spots evolved, which means it's very free to change up its spots quickly, and that's really advantageous because these butterflies, if you put them in a different environment, these spots perform different functions. They can either act as fake eyes, they can act to signal as like a confusion tactic to ward off predators. If they find themselves in a sandy area, they want to have no spots so they can blend into the sand. And the fact that it's this very simple gradient that's just like an on-off switch that causes these really complex regions means that you can have really rapid evolution where these spots will go away or reappear really fast.
So switching up the switches of a signaling node gives a modular and simple way to evolve.
So there's selection in action. These ones, for some reason, no one's really. I don't think people are really sure why these ones evolve in grassy areas, but it's pretty clear why these ones would evolve to look like, in sort of like a wooded area. These ones might evolve to look like an owl or a predator, and these ones would evolve to hide in the sand. It might be some sort of sexual signaling that's without without other predation concerns is preferred in these environments. But yeah, you have all three. They're easy to evolve. All the ones that don't survive get eaten up, and this is what's left. Yay, evolutionary theory.
Switches! Look at our spinal segments generate an hero. Built on a molecular clock. One single, one vertebra. One vertebra, one vertebra.
This is what I was talking about with Nigel, Nigel Braun of Nile Red, when he was talking about his clock reaction. I wish I knew more still about how this clock reaction works, because I don't remember. But it's like a series of it's a series of kind of like one signal causes another transcription, and then that signal causes another transcription, and that one signal causes another transcription, and then the cycle is complete, and it kicks off a gradient in the next region that does the whole thing again. And so every cycle you get a new little vertebral segment forming, which means that if you speed up the cycle, you'll just get more vertebrae, because you'll just keep going the cycle until you run out of cells. But if the cycle goes faster, you just, each one of these vertebrae becomes 3 or 4 vertebrae. And that's how snakes get all of their vertebrae. They just figured out a way to speed it up a little bit.
Speeding up its rate is snakes developmental cheat code. Better where a lizard's feet grow. They turn up distal aminos.
Yeah, it's distal. Distal is it tells you where your hands and feet should go. If you turn it off, just don't get hands and feet. So the crazy thing about that is that snakes still have the entire program within them for how to make limbs. And if you activate a snake embryo with the right distal protein, it'll grow up, not like a super functional arm, but it'll grow an arm. It's still in there. It's just been turned off. And that's I don't know, that's pretty cool, because you can just do one little thing, and like pythons sometimes will have mutations and grow little hind legs. But it's also that, like, according to their environment, what this means evolutionarily is that you can potentially have, like, snakes haven't necessarily lost their limbs. If something happens where a snake finds itself in an environment where it's advantageous to have some sort of vestigial limb, then whatever snake has a mutation that grows those limbs back, snakes will have legs again, because the program's all still there. And then, over time, snakes could grow their legs back. So it's quite possible that snakes have, like, lost and grown back their legs several times over evolutionary history.
Evo Devo. This is how we go from single cells to people. Every generation and in life primeval.
Probably not a lot to explain here, except that this little thing was very Bill Wirtz inspired. The way Bill Wirtz always has these, like, animation things all across the screen. I was using urbilateria, which is a hypothesized early bilaterally symmetric organism that gave rise to both us and flies. Bringing them back and having them wander around.
Variations and the sand beautiful. From Devo to Evo, larva to mosquito, patterns are resolved as the signals proceed.
Oh, I actually did put. Okay, so let's see how this circuit works then. So this is what I wasn't sure of before, but I forgot that I had actually written it down. So we have the original signaling protein from the mother. This isn't very helpful, because it's not really doing the whole thing, because it's more complicated than just activation. It must be activation in particular regions, because this one's only activated in the middle here. And then, yeah, this is too cartoonish to really get away with figuring out how it is that this works. So I'm sorry. This was enough work to draw as it was, so I didn't bother. This is how you figure out this stuff, is that you genetically manipulate your organism somehow so that the protein, that whatever protein usually gets expressed in these regions, you genetically manipulate it so that it's expressed along with this is a green fluorescent protein, which is sort of a standard tool. I can't remember where it comes from, if it comes from jellyfish or some sort of, like, microorganism, GFP. But it's a very common tool for figuring out where your protein is being expressed in an animal, because you just engineer it so that protein is attached to a GFP, and then you hit it with light and it'll fluoresce in exactly those regions. Morpholino is this is how they inhibited things in the days before CRISPR. Was that Truth Rationale Scientist? So, do you sing covers of songs but explain science? Yeah, Truth Rationale Scientist. I guess you're new here. Welcome. This is that's in fact what this channel is about. Yeah. Before the days of CRISPR, because now with CRISPR you can just send a little like targeted CRISPR in to knock out whatever gene you want. But back in the day, what you had to do was sort of put into the organism a little bit of RNA. I guess this is RNA that would bind with the messenger RNA as it was being expressed. So the genes would still be transcribed and like make all this RNA that's supposed to get turned into proteins, but you have a morpholino that's going all through the organism that keeps them from being turned into proteins, because it binds to them and then they can't fit into ribosomes. And that way you can see what happens to your organism once you've turned off the gene. CRISPR's made this sort of obsolete, I think, although I'm sure people still use it because there's lots of details involved in these things that they're usually better in one thing and worse in another. The Beagle is Darwin's ship, if you didn't know. Next time we'll see Banting, and you'll notice that this ship appears in the background. Because I was really into attaching the ends of old videos to the beginnings of new ones. Aesthetica in vivo. It's sort of okay Latin. I went to a bishop friend of mine who is very good at classical Latin, and I asked him, "Hey, I'm looking for rhymes for Evo Devo. I think maybe aesthetica in vivo is a thing that makes sense. I would imagine that it means some sort of thing like beauty in life. Could it work?" And he went, close enough. And so I decided to do it because it was just a good final line. Evo Devo. So there we go. That is Evo Devo. So what would I do at the end here? Do I want to do the end screen? I don't think I want to do the end screen. The end screen is just me talking. And we're just about out of time. And so I thank you for your participation. Hey, there's you guys again. We're going to go back to here.